Thank you, everyone. Welcome to today's EMSL Learn webinar on EMSL's Science Central, and we're so glad you could make it today. The EMSL Learn webinar series is designed to provide information to the scientific community on resources available at EMSL for your research in biological, environmental, and computing sciences. Our webinars are posted on the EMSL Learn webpage, or the EMSL Learn section of the, I'm so sorry. It's available on the EMSL Learn section of the EMSL website, and we invite you to visit that section to see past webinars. Our EMSL Learn webinar today, or I'm sorry, our next one is on May 7th, and we'll focus on X-ray photoelectron spectroscopy. More information will be provided through our social media channels and via email for all of our upcoming events and opportunities. So make sure to keep in touch with EMSL by subscribing to our email and social media channels if you haven't already. The links will be provided in the chat. Today's presenters include Soumya Manchakanti, who's an EMSL software engineer, and she will introduce Science Central. McKenna Detman, who's an EMSL data engineer, who will discuss data search and discovery on Science Central. And James Carr, who's also an EMSL data engineer, who will present on Science Central's modeling workbench and insight engine. There will be time at the end of today's webinar for questions. So we encourage you to push your questions in the Zoom chat so that our speakers can answer them during the Q&A. And so with that, we will begin with Soumya.
Hello, everyone. Thank you, Linda. Today we are, thanks everyone for joining the webinar today. So let's get started. So what is Science Central? So Science Central is a one-stop shop for all the users for all your research needs. It's a technological platform with broad capabilities for all the researchers, users, and scientific users. So the main aim of this webinar is to give you all a clear understanding of what Science Central is and what are its capabilities and where would Science Central fit into your research needs and processes. So what is Science Central? So Science Central is a unified research platform integrated with many different applications, each application built and tailored with its specific research needs. So as you can see here, so we have many different applications integrated within the main interface for seamless navigation for the users. And we're actively working to make sure all the integrated capabilities are in the integrated view to ensure consistent user experience across the platform. So using Science Central, users can submit the proposals, edit the proposals, track the statuses of the proposals, submit the metadata, download the data available and relevant to them, and use the downloaded data to do visualizations and data analysis, and also communicate and collaborate with the team members. So starting from the proposal submissions to data retrieval and data analysis, Science Central has broad range of applications to support your research life cycle. And all the tools of Science Central would require users to log in to access them. So let me walk you through the application now. So Science Central, as you can see here, has different applications, and also using Science Central, users can visit Proposals Calls website right here, which would have information related to the active proposal calls and also proposal submissions. And users can also visit EMSL Learn website, which is a one-stop place which has all the resources and video recordings for the webinars, presentations, and events at EMSL, which will help users to learn more about EMSL and activities happening around. And we also have a menu bar on the left that would help users to jump into applications when they are using one or the other one. So let's get started and understand the applications of Science Central a better way. So as you can all see here, we have 6 different applications of Science Central, each built with unique specific requirements. And before we get started into the applications and dive deep into them, we would like to share that today we all will be acting as research members of a LICTI group and will demonstrate how to navigate through how would research members or group would navigate through Science Central and interact with the applications throughout the research life cycle. Starting with, we have proposal management. Using this application, users can submit the proposals, edit the proposals, and also track the statuses. So let's get started on submitting a proposal. So let's click here, and using this drop down here, users can select the open calls that they are interested in and start a new proposal, or they can select the either of the topics that they are interested in. Once the proposal is submitted, it gets reviewed and user gets the status as approved. Using the proposals here and also the proposals in the quick links, users can get the status. So let's go ahead. And here, using this button, users can also create a new proposal and also start that. So once the proposal is submitted, it gets reviewed and user gets status. Right now, as you can see, I have an active proposal right now. So once it is approved, users might want to go ahead and submit samples to EMSL for further processing, which comes the laboratory information management system into picture. So using LIMS, we can submit the samples to EMSL. We have two different submodules, sample submission and L7. Let's talk about sample submission first. Using sample submission, users can submit samples and also shipments to EMSL. So here, as you all can see, I have multiple shipments which have been sent to EMSL. And if you did not have ever submitted, there wouldn't be any. Let's start and submit a sample. So now let's click add sample type. So how would I know that I have to click add sample type and start with it? So here we have a user guide that would help users to start with and give a clear understanding of what to be done, how would I submit a submission, or how would I create a shipping information, and how would I submit to EMSL. So let's get started and add a new sample type. So we'll be adding one of the Monet sample types right now. As you all can see, you all see an upload data file button here, as that EMSL will share the compatible files to all the users to fill and upload those here, which can be used to validate. Or users can also manually fill the data and review and submit them. So let's test one of these by entering the temperature. And I know that that's wrong, but let's validate and see how it goes. See, that's wrong. And it says me that I have many errors to be corrected. And using this the menu that we have, we can locate the right box or the right column that the error I am having at. So let's see where we have our sample temperature. Okay, so that's where I have my error at. And that's also a description that shows me what's the right type of input that I should have there. So checking that input, and once I have all these filled out and the errors are gone, now I have all the required attributes filled out, I'll now review and submit it. Once the data is submitted, I might want to fill the shipment. Oops, wrong button. Let's go back and do the shipping. So once the shipping so we can go to shipping and fill out all the shipping related details, like selecting the project, selecting the condition of the sample that's being sent, and review and submit that. So now the information is being sent and also shipment has also been started. What happens to this data? So all the samples once received by EMSL using the L7. So L7 is a tool that is accessible to the EMSL staff. So as you can see, which is a submodule of LIMS. So using L7, EMSL staff will be able to capture experiment and workflow metadata for the samples processed. And where does this metadata now sit? So considering that we have our proposal submitted, sample submitted, metadata submitted, and processed, where would the data now sit? So as you can see, there's a card that says data, which is the data platform of the Science Central. So data platform serves as the centralized repository for all the data generated by EMSL researchers, saying that So following DOE guidelines, we have ensured that the data is accessible to all the users, but users should be logged in to download them, balancing both accessibility and security at the same time. Makina will be talking more about the data platform and will walk us through how to download the data. But considering that we have proposal submitted, sample submitted, and data available. So once after the data is processed in L7, it is available for users to download in data platform. So once the data is downloaded using data platform, we now have the raw data. What can we do with that? We have modeling workbench, which has tools to empower scientists, researchers, and users, and any of these EMSL users to use those tools and do machine learning models, and they can do data analysis and visualizations using modeling workbench tools. James will be walking us through these tools in detail. And we also have Insights Engine. Insights Engine is also a set of visualization tools which will help users to create, saying that create meaningful representations or visualizations or analysis with the data that they have. And James will give us demonstration using one of the application or tool of Insights Engine. Last but not the least, we have Circles. Circles is a collaboration hub using which EMSL users can communicate and collaborate among each other. And using the collaboration hub, users can share, learn, and discover the scientific data as required. And currently, we are exploring Microsoft Teams as a collaboration hub and more about Science Central. Now I'm handing off to McKenna to walk us through the data platform and how to access EMSL data in detail. Thank you.
All right, thanks, Samia. So the first thing I want to do is take a step back and look at how the different applications within Science Central fit into the research timeline. So the research timeline in this case is basically the lifetime of a project with EMSL. So that would start with the proposal management tab. So this is where a PI could submit a proposal. Once it's accepted, they would need to enter the sample metadata. So this is what Salmi showed. Using that same portal, the EMSL staff would review the metadata. And once it's approved, the research group would send the samples to EMSL. At the same time, there's an integration between the metadata submission portal and L7, where those samples would appear digitally in L7 with a connection to the metadata submission portal. And what that means is as scientists record their activities in L7, once they're happy with the results, they've been reviewed and quality controlled, then those results can make their way to the data portal. So the data portal is where members of the project can find their own data. They can also find any data generated at the lab that's been released. So this will be what I show next is how to find relevant data and how to download it. Once you've downloaded data, you can use the Insight Engine and Modeling Workbench to do visual analysis, or in the case of the Modeling Workbench, machine learning and more advanced modeling. And James will talk you through that after my section. So with that, yeah. Okay, so to access data, this is the Science Central homepage. You can go over to the data card and it'll open it directly. You'll notice I'm not logged in, but we can still see data anyway. Right now we're on the All Data tab, which has project information at the highest level. So that's what we're seeing here. It's got things like the title, the abstract, the number of data sets. On the left, we've got project filters that can help us winnow down the projects shown here to just the relevant ones. We also have instrument filters if we're interested in results from a particular type of instrument. And then here we can also search. So I'm going to put in that same project that Saumi showed earlier. You can see it's a LICTI project. So in this case, I might be a member of the project who wants to get the most recent data available. So you'll see that we need to log in to actually download. So here I'm logging in. And now after login, I'm not seeing that little message saying we need to be logged in to download. Here are the data sets. We can filter and sort based on the columns. So I've sorted to the most recent data. If we hit the checkbox on the left, that'll give us the whole data set. But in this case, I'm only interested in specific files. So you can click on the DOI button. That brings you to a file picker where you can just pick the files you're interested in. I'm just going to grab 2 PNGs for this example. So I added those to the cart, and now if I click View Cart, we can see that those 2 files have been added. And then we can hit Download to actually initiate the download. You can enter a name. It'll help you stay organized in the next thing I'll show. We have 2 methods, Globus and just a direct download. I'll show both, but I'm going to start with a direct download. So now it's pulling the data off of the tape. So this can take a while. So I'm just going to close the modal for now. And I'll show you what the past downloads view looks like. I'll also talk about Globus here. So you can see I've already linked to my Globus ID. And I'll show you how to do that link here. Really quick, it actually just downloaded for me. You can see it's ready for download. So I get download again. But I'm going to show you how to create a Globus collection. So to do that, we need to link a Globus account. You can do that here. You can see mine's already linked. But if you hit the identities page, that'll be where you can find your Globus UUID. So it brings you here. I'm going to log out just to show the login process. So if we can go to the web app login here. And there are a number of ways you can use OAuth. I have a Globus ID, so that's how I'm going to log in. And here you just put in like your username and password, and it'll let you log into Globus. And when you log in, first it'll bring you to sort of your home page, which is not the identities page. So to get there, we actually have to go to the settings tab on the bottom. If we click the drop down, that's our ID. So that's the ID that we want to copy. And I just paste that over here. I already have it linked, so there's no reason for me to add it, but that's how you add it. And then once you have it added, you can create the Globus collection. So this is creating it for you. And then once it's created, it'll change from a create Globus collection to a visit Globus collection. This just takes a minute. Globus is a great tool because it makes it very easy to transfer between institutions. A lot of universities and national labs use Globus to move data, so it's pretty widely available. You can see that the files I selected were converted into a single tar file, so that's why it looks like that. And then on the right, you could transfer to any place you have Globus installed. It could be a supercomputer or even your personal laptop. Okay. The last thing I want to show is deleting. So I just hit the delete button, and it takes a minute to delete a past download, but we're working on improving the responsibility of that, or the responsiveness of that. Okay, so that's deleting now. And you can see it's gone now. So that's just a way to stay organized. Okay, so this is a Monet project, the Lichty project. So now let's say that rather than just our own data, we're interested in data across Monet. So we can use the Monet tab to find relevant data. Just like before, they're these same, well, they're different, but there are a bunch of filters on the left. You can also zoom in to focus on data that you're interested in. If you click the search visible region filter, you'll see the sample sets drop to 3. And now we have a search bounding box. If we zoom out, we can see the size of that box. It's pretty small. If we want, we can just hit reset map and then clear that to get all of our sample sets back. If we click on this top panel here, we can see that we have a number of analytics and a number of sample sets. The sample sets view shows just some metadata about where the samples were collected. Projects view looks similar to all data. And then on analytics, we can see the different analytic filters that are available. So for bulk density, we'll filter down a little bit. You'll notice that the sample sets won't go down until we actually check the box for bulk density to apply that filter. There you can see we've dropped to just 43 sample sets. But for this example and for James's example, we want to select everything except FTICR. We'll download that. Here you get a download summary that shows all the filters that have been applied. And this is a little different from the All Data tab as it just downloads directly rather than having to go off to tape. And so that's basically how you could get all of the Monet data. And in James's presentation, he'll show how to do some basic analysis on that. The other feature I wanted to sort of give a sneak peek on is the My Data feature. So this is something that's active now, but we're planning on adding functionality to it in the near term. So from Science Central, you'll need to be logged in to access this feature. And then My Data is available if you have an active EMSL project. So you can see it's fetching my active projects. It's that same active project that we've been using today. And inside, I've just got a couple of tar files. These are actually tar files that came from the data portal. In the future, we're planning to integrate my data with the modeling workbench and insights engine to make it so that you can grab data from the data portal, run analysis on it without having to download it locally. There are also a number of ways to access my data and these active projects, which there's some documentation on if you're interested. Thank you. With that, I'll pass to James.
Great. Thank you, McKenna. All right, so I'm James Carr. I'll be going over the modeling workbench and the insight engine. And if we go back to our timeline, we can see that at this point we're ready. We have data from the data portal, and we're ready to do some analysis on that. There's also the modeling workbench for machine learning. But in the interest of time, we'll just be going through some of the visualization features of the Insight engine. Researcher, you may want to do some machine. All right, so as a researcher, you might want to do some machine learning research, and that we have a few resources available on Science Central through the modeling workbench. Currently, we utilize Jupyter Notebooks pretty heavily. You can see we have the data science and the TensorFlow. And those are, for those of you that aren't familiar, Jupyter Notebooks they're interactive development environments that allow you to write and share code, and the documents for those are called notebooks. And I'll go into some detail about that and give you some examples as we go on. Just going to jump a little bit ahead here. And so it's also important to know that the data science and TensorFlow modules, they're running on our systems, so you don't need to download anything. You can just run it straight from the browser. Those systems that run those, it's also for lightweight processing. So if you have something more intensive, like you need to use some GPUs or entire nodes of processing power, for that, we recommend using Tahoma On Demand. And Tahoma is the name of the computing cluster that we have at EMSL for doing more advanced, intensive scientific research. So if you're approved to use Tahoma and you have a project that's approved, you'll be able to access that Tahoma On Demand box, and you can just click that and go in there. If you go in there, you will see that you'll get some pinned applications available. One is the cluster desktop, which is a desktop environment, kind of like your Windows or Linux typical environment. You also have the Jupyter Notebook or Jupyter Hub. You have our Studio server, and at the top, when you click interactive apps, it'll bring down a Dropbox, and you can see that we have other options available, such as Code Server, or you can use our Shiny apps as well. And we'll go back here. We also have the OpenML module that's going to be coming out. That'll allow you to organize and share your machine learning models. You'll also be able to share data sets with people on your team. And also we do welcome all kinds of feedback. So if you have ideas for additional apps or modules that would look great here, I can go through at the end and show you how you can give us some feedback, as well as report issues, things like that. So we'll go to the Insight Engine card. And at this point, you may want to upload some of the data that you received from the data portal and upload that to do some visualization or analysis. So if we click into that, you'll have two options right now. The Explore, which is a Jupyter Hub environment, and the Map tool. And that's the multi-omics analysis portal that was also developed here at PNNL. And there's another EMSL Learn webinar specifically about the Map tool. So if you want more details on that, how to use it, things like that, be sure to watch that. For now, we'll go into the Explore Jupyter Hub module. And when you go into the Explore module, it will take a second to start up, but it's very fast, and you'll be presented with a screen that kind of looks like this. So from here, we have the data that we downloaded from the data portal, and we want to upload it, and it'll appear there on the left pane. That's kind of your working folder. So we'll start by creating a notebook, and to do that, you will click the Python 3 button there under Notebook, and that will create a notebook file for you. And that's kind of where you'll put your code into the cells there. And then you can go up and click the upload button there at the top, and that will give you a file picker, and you can select files from your local machine. They'll be uploaded, and you can see those on the left pane there. Those are a couple of CSV files that we got from the data portal. When McKenna showed you the different filters and click the download button, it'll give you these CSV files that are kind of an aggregate of all of the data based on those filters, as well as some metadata. And you can see we have some things like ecoregion, soil type, vegetation, and then we have our actual numeric data that we downloaded. So you can view those in there. Um, and we also have a drag and drop feature. So for this case, I wrote some code already, so I didn't have to do it during the presentation. And I'm just going to drag and drop that code file into the left pane there, and you can see that pop up. I called it plotter because basically we're going to take that data and make a bunch of plots so we can do some visualization. So I take the first piece of data there and copy that over to the notebook cell. And all that does is basically takes those two CSV files and combines them together so we get something called merged data CSV. And I'm also selecting specific columns to use. So you can click the play button to run that, or you can press Shift Enter and it will run the cell. You can see that merged data CSV is created, and we can open that to look at it. Once that looks good, we can go back and grab another piece of code and copy that over. And what that does is it basically, we're going to create plots for different columns, and the x-axis is going to be pH, total carbon, nitrogen, and sulfur. And we're going to compare that with ecoregion and the soil type. So we're going to create a whole bunch of plots that basically permutate through all of these columns and generate plots. So once we pressed Shift Enter to run that, it should start populating at the bottom. You can see it starts to output. And then on the left pane, you can see that these image files have been generated. And if we expand the pane, we can actually see the full file name. We have a bunch of PNGs. And they also get put into the output window, and we can scroll down and look at these graphs that were generated and do some analysis on that. So once you're finished with that, you can, so actually in the future, we're going to have an ability to select multiple files and then have it zip them automatically, and you can download. But for now, I created a little piece of code that does the zipping for me. It takes a plots folder and zips it up so that we can download a single file. Otherwise, we'd have to download each image individually. So here I'm creating the plots folder, and I'm just going to click and drag the new images over to that folder. And we'll run that cell to create the zip file. And you can see we have plots.zip, and you can right click that and go to download. And that will download to your system, and you can choose that from there. Let's see. And oh yeah, also, as I mentioned, the files will remain in this workspace between environments. So if you want to go back to modeling workbench and do some TensorFlow using Jupyter Hub, those files will remain there, and you can come back to the workbench, or the Insights engine, and access those same files. So those don't go anywhere. In the future, as we implement the active project directories, entire teams will be able to collaborate on the same set of files. So you'll be able to share that and collaborate with your team members on that. Moving back to the main page, I just want to go over some of the help menu options. Here we have a user guide, and that goes through the different modules of Science Central, how to do certain things. There's also how to create a Globus account and all of that fun stuff. We also have the report and issue button, and that will basically open your email client, such as Outlook, and you can send an issue report. We also have the send feedback form, which I highly recommend you all do today, and you can access that from the Science Central main page. So you can send feedback about a specific module, send us your suggestions, anything like that. We'd love to hear back from you. And I think that concludes my section. So thank you all very much for listening. We'll move on to the questions.
Thank you, James, and thank you, Sonia and McKenna. Okay. So let's start with some of the audience questions. And a reminder to those in the audience, if you do have questions, feel free to drop them in the chat and we will capture them so that our presenters can answer them for you. So Sonia, first question, can I use Science Central even if I don't have an active user project?
Yes, considering the broad capabilities provided by Science Central, I would encourage every single person right here in this webinar and watching this webinar to create a Science Central account and come and just explore the applications that are available, because even if you do not have an active project, you can still browse the available data, download the data, and play around with the data using the tools from Modeling Workbench and Insights Engine. And even if you're not interested in using our data, you can still use the Jupyter Hub notebooks to still have productivity on your visualizations and data analysis if you're interested in. Yeah, hopefully that answers your question.
Yeah, great, thank you. So this next question is for McKenna. Is there a limit to how many files you can download from the data portal at once?
Yeah, so there's currently not a limit on the number of files. There is a size limit. I believe it's 100 gigabytes, but if you need to download more than that, you can do it using Globus. Like I mentioned, Globus is really good for moving large amounts of data between sites. So that's the preferred way to move those really large downloads.
Okay, and as a tag along, since you mentioned Globus, can I use Globus to download to my personal machine?
Yeah. So one of the big use cases for Globus is to move between sites, like I mentioned. But you can also use it to get large amounts of data move between some site and your local machine. You can do that if you download and install Globus Connect Personal, and then with that you'll be able to move it just like it's any other endpoint.
Okay. Have a question about Monet. So can someone tell me a little bit more about the Monet data, how to access the data, what you can do with the data at this moment, and any future updates that we can expect for the Monet data that's currently available? Is anybody able to take that
Yeah, I think
question?
James.
Yeah, I
Okay.
can answer that. There's constantly new data coming in for Monet. We right now there's fiscal year 23 sample data that's already in the data portal and available. We're working on fiscal year 24, and then after that, of course, 25, and it'll just keep coming in. We're also adding some Thousand Soils project information and new data types as they come out. There's plans to do something like metagenomics is coming out very soon. And then site-specific data, so you'll be able to get images of the sites where the samples were taken and stuff like that. So yeah, it's a constant flow of updated information.
Okay. How is that data going to help the research community though? Like what, is it just downloadable or can they do some kind of analysis through Science Central with that Monet data?
Yeah, so as I was showing in my demo, you can do some visualizations on the data that's already there. That's just the basic metadata that's on the Monet tab. You can, if you choose, download specific instrument data like the raw FTICR files. If you're familiar with that, more advanced users can get those much larger files and do any sort of analysis. It really depends on what your research question is.
Okay, fantastic. Thank you, James. And I have more questions for you too, James. So can I share the Jupyter notebook I made with others on my team?
Yeah, so when you go into Jupyter Hub, you can right-click the notebook file and choose download. That's probably the easiest way. In the future, we'll have the active project directories so multiple users can log into the hub and see the same directory and the same files, as long as they're part of that project.
Okay. And another, what is the difference between the notebooks, like TensorFlow and data science? Talk a little bit more
Yeah,
about that.
yeah. So each of those, they all rely on the same Jupyter Hub, but with different environments. So these are things like the pre-installed packages, like the TensorFlow environment will come with TensorFlow and Scikit pre-installed. If you're good with Python packages and how to install those, you can create your own environment and have a fully customized setup. So yeah, it's just pre-installed common packages that truly sets those apart.
Yeah. Okay. So I'll asking the audience, are there any further questions? I can see that I think we've gone through all of our audience questions so far. I know some of you out there are probably trying to think of how to formulate that question. So we'll give you a little bit longer, but if no more questions come through in the next, we do have a question, woohoo. Can I upload different metadata types? It's a great question. Anybody want to take that?
Yeah, I could take that. Yeah, there were, I think the question is about the metadata submission portal. There are a number of types supported, and you can go and check that out yourself. We also have metadata point of contacts who can help you fill out that metadata as is relevant for your project. So depending on what type of proposal you submit, there would be different types of samples that you would need to send to the lab. So that metadata POC will help you figure out exactly how to fill that all out.
Great. Is there any other questions? If there's not any more questions that folks can think of right now, you are always welcome to send questions to folks' way via email. I believe that everyone's email has been provided in the chat links. Yeah, you can go ahead and email folks for additional questions. We did get another question that came in. Can you provide a ballpark timeline for the direct connection of data to the bench?
Yeah, that's a good question. It is one of our priorities. I think either FY 25 or 26. But we do recognize that it's an important connection to make because we want to make that process of grabbing data and then doing analysis on it as smooth as possible, especially for larger data sets. So yeah, it's definitely something that's on our roadmap.
All right. Okay. And I believe there's another one. I may have missed this, but are there standard notebooks available?
Yeah, yeah. So in any of those Jupyter Hub instances, there's a way to open a standard notebook. If you're interested, I can just shoot me an email and I'll show you how to do that. But yeah, there you can open a standard notebook in any of those environments.
Mhm. And your email would be mckenna.detman@pnnl.gov. So hopefully, if anybody has questions about that, you can email McKenna directly. Okay, I think we're going to go ahead and call it so that folks can get a few more minutes of their lunchtime back. So thank you again to our presenters today. This was great information about Science Central, and we hope for more webinars like this as Science Central continues to add additional capabilities. We are going to keep our user community informed of those updates, and so we look forward to seeing you all back here again for a future webinar. So, and thank you to the attendees. Have a great rest of your day.